today's topic is Behold His Beauty, Design of the Human Visual System. So if you're coming to this talk, we have to use some words in this talk. So is everybody comfortable with the word eyeball? Okay, eyeball. All right. And then and we're going to look at some pictures in this, and we're going to look at some pictures and features of the eyeball. And for some reason, some people get freaked out when they look at an eyeball up close and stuff like that. It's like, oh, I can't stand it. So if you're looking at a picture of an eyeball or a little bit of a segment of an eyeball, and you get that queasy, woozy, quoozy feeling, please vomit sideways, not forward, on me, and share the love with the person next to you on that. After Dr. Perdam's lecture, you'll just be sharing some symbiotic bacteria with them on that. Design of the human visual system. Well, remember this hypothesis. Worship should be the normal response to science. Hasn't changed. We're fearfully and wonderfully made, and now we all know to say the whole verse, I will praise you because I am fearfully and wonderfully made. Everything is made. Made. Boy, pound this word into your mind. Made. The Lord has disclosed himself in Romans 1 by the things that he has what? Made. Even his eternal power and Godhead, so that they are without excuse. I wish we had never lost the words intelligent design, because everything is a design argument. And please, let me encourage you, start looking at your human body like you would look at any other sophisticated designed entity. You can only really understand it as you understand the principles of design. Oh, we need to get the rest of our creationist biologists on board with this. You can only understand it properly as you understand how things are designed. So we're going to look at some of the functions of design, and if you want to write it down, it's nowhere on the slides, but designed complexity is this. Hold up your hand. It is multiple parts working together for a purpose. That's what complexity is. Multiple parts working together for a purpose. We could put a mathematical probability on this. We could do that. But multiple parts working together for a purpose is biological complexity, and that has only been observed in any other area from an intelligent source. Natural forces alone, gravity, wind, rain, all those things can't even produce two parts working together. Not to mention all of the parts we have working together. So biological complexity is what? Multiple parts working together for a purpose. And you know what? When people see multiple parts working together for a purpose, inside their mind is saying designed, designed, designed. And the next word that you want to use when you're talking with them, this is all extra, it's not even a talk, is the word fit. Fit. Things only fit together when they are designed. It's the perfect fit of things which shows the evidence of design. Now, I talked about human reproduction today. Did everything, even at the cellular level, just fit perfectly? It fits like a hand in a glove. And we didn't even talk about the external anatomy, which fits like a hand in a glove. I knew someone would get a chuckle out of that. Sorry. It does. It fits like a hand in a glove. But anyway, it all fits together. And when people start seeing how things fit, they know they only fit by a design source. So you use the word fit. Birds' wings fit their environment. Fish gills fit their environment. These things fit. And the things we're going to talk about on the eyeball are fit. And when people start hearing how things fit, they will start praising the Lord because they'll know that they are fearfully and wonderfully made. Well, this is what we're going to talk about. This is a painting of a cross-section through an eyeball by this man named Frank Netter. He was also a medical doctor, but he was a great artist. And everybody who goes through medical school studies Frank Netter's pictures. And this is a cross-section through an eye. You can see your cornea right up there. You can see your lens here. You see the iris with the pupil there, the retina in the back. We're going to talk about all of those things. This man is now dead. I heard him interviewed on ABC News by Peter Jennings, who's also dead, before he died, and I don't know whether he was a believer or not, but he said in his interview, I find it hard to believe that all the things I've been painting for all these years could have come about by chance. And that just popped out. I'm surprised ABC didn't edit it out of the interview, but it was in there just to say. I want to give him credit because I'm going to use some of his pictures. Now, visual system, light. Dr. Mason already mentioned this word, photon. A photon is the smallest unit of measurable light. It may be a particle, it may be a wave, it may be a wave-like particle. I don't know, but they travel at obviously high speeds, which is the speed of light. There we go, traveling at the speed of light. You see various colors reflecting off the screen, and those are variations of absorption. And light is just pure energy, and you're able to detect the smallest unit, a single photon of light. And what we're really after here, since it's a designed mechanism, is we need something that actually has an ultimate purpose. And the purpose is to get information. That's what it really means to see, is to get information. And so I'm going to mention that over and over again. The purpose is to eventually store information. So you need a mechanism which is able to sense, detect something. Light is reflecting off this screen. It's coming into your eyes. You have a detector mechanism that is called your eyeball that detects the light. And then you need another system to make sense of the data and convert that into information. That is your brain. You have a visual system which consists of your eyeball and your brain and all the muscles and everything which make those things work. This is not a talk on the eyeball, which everybody believes that it is. It's on the entire visual system and how these things must work and function and operate together. And I use the word miracle because it is a miracle, because it's going to show that all or nothing unity in which all the parts must be there as nothing's going to function. Well, the development of the visual system, there are two explanations. Supernatural creation, all of these parts fit together for a purpose, i.e. all the features of design because it had a real designer. Or the naturalistic evolution explanation, in which a series of random genetic mistakes were guided by natural selection, whatever that is, culminating in a highly complex system. Those are going to be the two explanations. You decide at the end of this talk which one of these best fits the data. Well, one person, his name is Richard Dawkins. Man, what a treasure trove of quotes for us, because he states what's on his mind very candidly. In a book called The Blind Watchmaker, he was lamenting that people have a hard time understanding evolution. He says, it's almost as if the human brain were specifically developed to misunderstand Darwinism and find it hard to believe. Wow. You know, it's like that. Because the Bible says these things are clearly seen right there. The conclusion that our brains seem predisposed to resist Darwinism stems from our great success as creative designers. Our world is dominated by feats of engineering and works of art. We are entirely accustomed to the idea that complex elegance is an indicator of premeditated design. And to that I would say, no duh. You know, when we see complex design and we conclude there was a designer, that is a totally reasonable, totally rational conclusion. It's the irrational conclusion which sees complex design and believes that's all an illusion of a designer. He goes on to say, this is probably the most powerful reason for the belief held by the vast majority of people that have ever lived in some kind of supernatural deity. The Bible is true. It is clearly seen, and people in any age and any generation see it. Of course, Dawkins thinks that the vast majority of people that have ever lived are all wrong, and he is right. But we're going to see that the vast majority of people are probably right. Well, your eye, going to begin development about the 4th week of pregnancy. Already those tiny little eyes are going to begin forming, and it is going to develop from 3 different layers of tissue in this tiny little embryo right here. This is a picture of an embryo, and right up here on the top of the head where it says lens placode is where those eyes are going to begin developing. Here's a drawing of that, and this is the developing little eye on that tiny, tiny little embryo. Now, the embryo shows all kinds of features here. It doesn't look totally human, it looks different. And those things are being exploited in evolutionary textbooks, and we'll talk about that in just a second. But the 3 different tissues are guided by genetic information, which has 1 layer of tissue cup in and infold on itself. Another layer of tissue is going to have a little piece bud off right in here, and that's going to become the lens in this tissue. Another layer of tissue, this is really cool, is going to extend out. It's going to make a scaffold for holding that lens in place while it is forming until tiny little ligaments can completely encircle it and hold it into place, just like you would use a scaffold to hold something up on this building until its structure is put in. And then when the structure is put in, you take down the scaffolding, and right on cue, after that lens is in place, the scaffold breaks down, goes away, because if you had a scaffold right behind your lens, when light came in, you would not get good information. So you have to have it out. So it's built up, utilized, taken down right on cue. Now you see that little embryo right there. This is what a biology textbook. These pictures were taken from this. This is a typical one. And it says, it's kind of small for you to read, for example, the human embryo has a well-developed tail and also a series of gill pouches in the pharyngeal region right here. Consequently, it looks very much like an early fish embryo right up here. And it says human and fish embryos resemble each other because human beings share a common ancestor. Wow, that's what it's being told. The problem is none of those factual things pointing to that are correct. This is not a tail on a human embryo. When that baby is being laid down, the backbone is put down first, like a scaffold, and the rest of the body grows into that backbone. And this embryo is just in the process of growing to it, and this portion will never disintegrate. It's not a primitive tail. In fact, the baby is going to grow all the way up right here to the very tip, and that is the coccyx, the bone that you are sitting on right now. It has muscles which anchor the pelvic floor of your body. It has muscles which anchor certain other muscles which are keeping certain orifices closed in your body right now, which I'm glad are really functioning for you. It is not a useless thing whatsoever. And over here, these little lumps are not gill slits, they're pharyngeal pouches. And they're going to develop into structures in your neck. They're going to develop into your thyroid gland. They're going to develop into your lower jaw. They just are folded up, and people look at them and they think they're gill slits, but they're nothing like gill slits. So their whole premise is wrong, and consequently their conclusion that we share a remote common ancestor is wrong. Well, there we go. Let's get back to babies. Baby's going to be born. Baby's eyes are going to be 75% of adult size already, 90% by the end of the first years, and very, very little growth after puberty. Now, why does baby have these big, big eyes? It's because they look so cute. I knew it. It's just because they look so cute, and they kind of melt your heart on their first birthday. Well, that's a good reason, but the reason why they have these big eyes is because they function for a purpose. Baby needs to be taking in data adequately from both eyes right from the beginning, so the neurological tracks from the eyes back to the brain will develop properly. If one of baby's eyes is turned out slightly, the doctors will do what? Patch the eyes alternatively so both eyes are being forced to be used so the data comes in and the neurological tracks lay down. If you don't do that, the baby will be blind. And so baby needs these large, big, functioning eyes right from the very beginning. Well, if you were to take someone's eye out, so if you donate your eye to science, this is what we'll do. We'll cut this eye in half, we'll stain it right here, and we'll show everybody all the features of the eye. We'll show that your cornea doesn't stain, it's nice and clear. We'll show the lens, we'll show the retina, and these are all the structures of a real human eye. So that should entice you. You want to donate your eye to science so we can cut it in half and show your friends what your eye used to look like on the inside there. This is what a real human eye looks like in half. But there's more. There's more to the eye than just the eyeball. Look at these other important structures called the eyelids. Now the eyelids play an important role to vision. I used to have patients that would come in, only had two when I worked in the emergency room, and they were involved in automobile accidents and they had their eyelid ripped off. Now, ooh, ouch, that's a bad deal. When you get your eyelid ripped off, don't throw up that away. All right. It's an ophthalmologic emergency. You need to cover that eye or the eye will dry out. That's because your eyelid provides an important function. It has all different kinds of glands in it. One of them, called the meibomian gland, secretes a thin layer of oil, which keeps the watery tears from evaporating away too quickly. And also, every time you blink, 6 to 7 times a minute, it provides a layer of lubrication for that eye. So it's very, very important. It also mentions that that oil fills in any irregularities on the corneal surface, making it perfectly smooth so that the light coming into your eye is not distorted, because distorted light will give you distorted what? Information. So you want all of that working together perfectly smooth. And this is what it looks like if we were to have a drawing of this, a cross section through the eye. So eyelid, you would look at this from the side. Even something as small as your eyelid has all kinds of details in it. The outer skin, tiny little muscles on the inside, you're looking at them from the side right there, which allow you to curl your eyelid up. There's even a muscle which goes up here to your brow, which allows you to retract the eyelid. There's a piece of dense connective tissue right in here called the tarsal plate, which gives your eyelids some rigidity. And little boys are able to take their eyelids and flip them up, and they kind of stay in place. They chase the girls around on the playground. It's like a primitive mating ritual or something like that. It happens in all the cultures and everything. It's funny, it works. But anyway, it has all of that. And inside this tarsal plate is a gland which makes that oil, which is secreted and goes under here on the part of the eyelid that touches your eye. And this is what it looks like in real life. This is a cross-section of someone's eyelid right up here. You can see all of those structures. Here's the muscle tissue, the outer skin. You can't see the tense connective tissue. It's washing out here a little bit, but there it is. Here are the oil glands. Here's the pore for the oil glands. And you see all of these structures right inside an eyelid, which would have had to develop by numerous successive slight modifications. Lots and lots of details, even in an eyelid, which is absolutely essential for your eyes to function. Now, one last question. Is this eyelid from a man or from a woman? Well, this is clearly from a man, because if it was from a woman, there'd be all kinds of makeup caked up here on the outside of that you would cut through. So this is from a man right here. All right, let's talk about another important gland, the lacrimal gland. Lacrimal gland is going to make your watery tears. They lubricate your eye during blinking. What's really cool about these is they not just have water, they contain, as it mentions there, antibodies and lysozyme. These are protective features in your tears which will kill bacteria on contact. Lysozyme destroys them, and you have antibodies there. And what's also cool about human tears is they express something called enkephalins, which are natural opiates. These materials expressed are in the same category as morphine or Demerol. And these drugs give people a feeling of euphoria, and not only taking away the pain, but the feeling of euphoria is what's so addictive about them. And so, as human beings cry, these natural enkephalins, natural opiates, are being released. And only human beings cry emotional tears. Animals don't. Your cat doesn't cry tears of joy when you come home. And your dog isn't crying when you leave. Only human beings cry these emotional tears. And that's why after people cry, they usually have that big sigh and they feel better. Now, I wonder how long it took numerous successive slight modifications to get something, and why this would even come about. Why would evolution ever put this in? Or is this something from a gracious, loving God who would put something like this even in our tears? Well, here's what that lacrimal gland is. It's a cutaway up here, right up here on the outside of the picture. Now, I do this talk in Texas, and I actually have to tell those people down there, this is not normal right up here. This is a cutaway view of the head. And what's interesting is these tears are made by this gland. They go through tiny little ducts. They sweep over your eye, and they're picked up through two tiny little openings right here. And if you look in the mirror really close, you can see those little openings. They're called puncta. And the openings are actually surrounded by very, very tiny little muscles. And every time you blink, the muscle contracts, and when it opens up, it sucks the tears off. Sucking the tears off of your eyes. They go into a little tube. They drop down here at the back of your nose, and every time you make this sound like that, you swallow the tears down. They go down the back of your throat, into your stomach, and you recycle them over. Eventually, they're made into new tears and to other things as well. Now, these tears have to be made at just the right amount so that your eye stays lubricated, and they have to be absorbed at just the right rate so that you don't have tears flooding down your face causing infections. This is what happens with people who have this apparatus infected right here and they can't absorb the tears. All of that has to be produced and absorbed at just the right amounts. And these are just accessory structures for your eyeballs. Well, now we get to the eyeball itself. Here's the main feature, the cornea. It's the clear part of your eye which is going to do the major focusing of the light of your eye. 45 diopters, most of the focusing is going to be happening by the cornea. Another cool feature about it is the cornea is invested, the outer layer, with all kinds of pain fibers. And that's why when you get something in your eye which could blind you, you just can't ignore it. It forces you to stop what you're doing and get the thing and get it out of your eye, because pieces of sand and sawdust and stuff like that eventually will make you go blind unless you take them out. So pain fibers are actually a blessing here to let you to have you fix things that need to be fixed. Well, here's a picture of a human cornea right here. This is the clear part of the eye, and I put this picture up because it is opposed to an optical grade piece of glass in this contact lens right here. And even in this room here with just digital photography, you can see that the clarity of this glass doesn't even match the clarity of the cornea. The cornea is essentially crystal clear on a normal healthy cornea, so none of the light is distorted. Well, how do you get a crystal clear cornea? The cornea is actually made up of multiple layers, just like layers of plywood. And the layers are stacked right on top of each other. They're pressed together, and they're kind of glued together, just like a piece of plywood. And what happens if you leave a piece of plywood out in the rain? It swells up, and the plies start to pull apart. Well, this is a cross-section of a human cornea. Here's the outer layer. Has all of the pain fibers. These are all the layers that are stacked up on top of each other. The tiny little blue dots are the nucleus of all of those cells. The little breaks are because when you take tissue out of human body, it starts to decompose and it breaks apart. But in real life, they would be stacked right on top of each other, just like a piece of plywood. Now, how do they stay so close together? Well, inside the cornea, on the inner layer, there's a layer of cells, and that layer of cells has several functions, one of which is to pump sodium molecules out of the cornea. And every time it pumps a sodium molecule out, a water molecule follows it. So it essentially pumps water out of the cornea, keeping it all those layers very, very tight together and keeping them tightly packed. If those break down, such as in the picture here you see of this person who has a disease of their cornea, the cornea will begin to swell up and become cloudy and white, just like the white part of your eye. Because it's made out of the same materials as the white part of your eye, but it's that special layer of cells which have this special pump on it, which pumps it out. And so here we have that human eyeball. If you were to take a little slice of the cornea, blow it really big under a microscope, you'd see this outer layer of cells, some of the fastest growing cells in your body. So if you get a scratch on your cornea, it actually heals very quickly. Here are the densely compacted layers. And here is the inner layer of cells, and this is what they look like magnified many, many, many times, that pumps the sodium molecules out, keeping this essentially dehydrated and packed together perfectly clear. And you need all of those features just to get a clear cornea. Well, other design features there in order to keep that cornea completely clear is it can't be covered with blood vessels. You wouldn't want blood vessels growing all over your cornea because that would distort the light, which would distort your what? Information. But a cornea is a living tissue. It needs oxygen. It needs nutrients. Where does that come from if it's not covered in blood vessels? Well, it comes from a fluid called the aqueous humor. The aqueous humor is actually made by a tiny little organelle right in here, right behind the cornea. And how does it make the aqueous humor? This is kind of cool. Blood is pumped from your heart. It eventually is pumped up to your eyes through tiny little capillaries, and it's pumped under pressure. And the pressure is just right so that as it flows through the ciliary body has tiny little cells in it with tiny little holes which function like a screen. And the holes in the screen are so small that even a red blood cell will not fit through it. But water molecules will. Water with dissolved sugars, with dissolved oxygen in it, water will flow through, but the holes are so tight that the proteins and the red blood cells stay behind. So it's a filtrate of your blood, and it is crystal clear. Crystal clear. And it is pumped out, and it circulates around behind the cornea. This is your cornea. It circulates around in this space. It nourishes the cornea and nourishes the lens with the things that are in it. And this is what it would look like, really blown up on a picture. This is your cornea. This is the colored part of your eye, the iris. Here's where that ciliary body is. It filters the blood, makes the aqueous humor, flows out in front of your lens, circulates around in your eye behind your cornea, does its job, and when it's done, it is absorbed through thousands and thousands of tiny little holes which are in the perimeter, right where the colored part of your eye attaches, goes into those holes, picked up by your venous system, and goes right back into your blood circulation. And that is produced at just the right amount and is absorbed at just the right rate to keep your eyeball pumped up, firm enough, but not too firm that it crushes the retina in the back of your eye, which is the result of a disease called glaucoma, which can kill the back of your eye. And you can tell if your eyeball pressure is about right. You can take your thumbs, and I have to tell people in Texas, close your eyes, but you could close your eyes, and you can put them on your there, and you can push it, and it should feel about the firmness of a grape on there. Not soft, not too hard, not hard like a rock or anything like that. And so that is how you nourish this eye, through a superfiltrate of blood. Now, I wonder how long it took numerous successive slight modifications to get that right. Well, it not only nourishes the cornea, it also nourishes the lens. And this is a lens from a real human body. And you can see it actually magnifies this grid. Your lens is used for your nice, fine focus. And in a real human body, that lens would be crystal clear. But once you take something out of someone's body, it starts to get cloudy. Some of you are getting older, and your lens is getting cloudy. That's called a cataract there. But with young people, it's generally crystal clear, and it's very, very flexible, so that young people can look at things very far away. They can put their hands really close to their face, and everything stays in real quick focus because the lens can actually change shape. Now, some of you haven't seen your hand up close like this for a decade, but that's because your lens is actually getting kind of stiff and old because you're getting stiff and old. And so that is why your lens doesn't do that. But what allows the lens to flex like it does? Well, if you were to look inside the lens and magnify that hundreds and hundreds of times, you would see the lens is made up of tiny little proteins called crystallins, and they are stacked on top of each other in a tight, orderly fashion, and they have a little joint. Can everybody see the little joint between each other? And that joint allows those little proteins to flex and to fold and allows your lens to change shape. It doesn't just happen, it's designed to happen by all of these little design features right inside your lens. And you have all the information to code just to make these tiny little crystallins in your lens. Okay, this is in here. This is like a little intermission. I put this in because there's kids in the room, young and old ones, and I just want to show you different kinds of eyes here of these different kinds of creatures. And what's interesting about this is the DNA to code for all of these different kinds of eyes is essentially the same. It's how the DNA, how the information is used with another layer of information on top of that, which allows you to get all of these different types of eyes that I'm going to show you. So look at the little eyes of that little salamander right there. Those are kind of cool eyes. And here's the eyes of a creature called an eye. And it's called an eye. It's got neat looking little eyes. And here's a picture of a tube-nosed bat's eyes. Another type of eyes that the Lord has made. Eyes from obviously a dragonfly right there. Eyes right here of a little crustacean. I love them because one eye is looking up here and one eye staring at the camera right in. We have eyes obviously of the fruit fly, the poor misaligned fruit fly, which is supposed to prove evolution but shows that fruit flies have always been fruit flies on that. We have the eyes of this little baby fruit fly in the incipient stage right up here as they're being formed. Flies of a spider, kind of a cool eye right there of his reflective little eyes. Eyes of a tree frog on that. This is the eyes of a mantis shrimp. We can see, we can see red, greens, and blues. This mantis shrimp can pick up 12 different primary colors. Some people suspect this mantis shrimp has the best eyesight of any animal in the animal kingdom, with those looking eyes, and supposedly a primitive creature there. So eyes of these two little creatures right here, kind of loving on each other. On that, the orangutan and the dog, and that poor dog was orphaned when he was just a puppy. And mom was killed, and orangutan took him under his wing, and now these two are like the best of friends. Aren't you looking forward to the day when the curse is gone and the lion will lay down with the lamb and we're going to see this creation for how it was meant to be? Oh man, we're going to see this stuff. It's going to be glory to see all of these things. Are these eyes of this little insect right here? There's the eyes of this caveman, that everybody sees on that. But it's not really a caveman. It's a man made up to look like a caveman. Or the eyes of this gal who was on the picture of National Geographic with her stunned eyes, a gal from Afghanistan who thought the photographer was going to steal her soul. And that's why she's so afraid when she had her picture taken. But she had a hard life, because here's a picture of her 30 years later, and the eyes more of a woman who has seen some of the hardships of life. And these are the eyes of the people we need to take the gospel to. Well, now you've come to the ophthalmologist or the optometrist. They've stuck your head in that device, said, put your forehead up against here, and now they're shining that bright light in your eye, saying, don't blink on that. That's what we do for fun. It's kind of like mini torture. So when you stick your head in that device and we shine it in the back, we're looking at the back of your eye, and this is what it looks like. This is a healthy tissue. This is healthy retina in the back of the eye. Here's your optic nerve. Here's a tiny little artery coming out, tiny little vein. This is your macula, and this tiny little spot there is your fovea. When people get macular degeneration, they get a disease with this part of your eye. All of the light and everything you're seeing is being focused down to that tiny little dot in the back of your eye, about the diameter of a pencil lead. Everything is being there. Now, how you're taking in other things, your eye actually moves very, very rapidly in tiny little movements called saccade, which is moving that area of hard focus around very, very quickly and imperceptibly to you, so that you can take in more stuff than what is in hard focus. And here is what it looks like right here through a head. So if you donate your head to science, this is what we'll do. We'll take the top off, look inside, see if there was really anything in there, and we will then show it in medical lectures. Now, I don't put that in there just to, be gruesome or anything. It's because there is a feature in the back of your eye which really is important, and you can't see it from the outside here. You have a layer of black pigment behind the retina on both eyes. Black pigment. Now, why would you have black pigment there? Well, think back to when we used to use real cameras with real film. You opened them up, and what color was it? Black. That's because light coming into the camera, you wanted to strike the film and develop it at one spot and not be reflected all around inside, so it was painted black. Well, we want a photon of light to come in through your eye, strike your retina on one spot, send that piece of data, and we want that photon to go through that and hit this black pigment and stop and not be reflected around. If light were to be reflected and scattered inside your eyeball, you would not get good information. You would get confusing information. So here's a cool design feature which nobody ever sees unless you look at it from the top of someone's head here. So that's why it's in this, that's why this picture is there. Now, for the next 5 or 6 slides, we're going to get smaller and smaller and smaller and smaller to tell you everything what's actually happening down at the back of your eyes. Because as the pastor said, God is in the details, and you must experience the details to fully appreciate God. So that's why we're going to do it. So this is your retina magnified really huge. You have about 250 million photoreceptors in the back of your eye. These tiny little brush-like cells, can everybody see them here standing up on end? Those are the photoreceptors. Those are the cells that are actually pulling in the light. All of the little blue dots above them are stained blue. Those are all of the neurological junctions where the cells come out and they make a junction. Are they making a lot of junctions? You bet, because you're moving a lot of data. So we're going to focus on these brush-like photoreceptors in your eye. You look at this little black layer right here. Guess what that black layer is? That's the black layer I just showed you on the back of the eye, which is stained black because it is black right there. Well, you have 2 different types of photoreceptors in the back of your eye, and we've all heard about them, cones and rods right here. The cones, and this is what they look like magnified even bigger. This is a cool photograph. It was able to take away all of the other tissue around the cones and let you see just the cones themselves. Now, cones are useful for seeing really fine details and also for seeing colors. So as shown here by this picture here, you're able to pick out all these different colors in because you have these cones. The other photoreceptor is a rod. And that's another cool photograph where the other tissue has been stripped away. And you're able to see all of these photoreceptors, the tiny little rods, and rods enable you to see gross movements, large movements, and they're able to enable you to see in the dark, black and white, very, very well. So you have both cones and rods in the back of your eye. Well, what makes up either a cone or a rod? So if we were to take one of those cells and magnify it even bigger, we'll look inside what's in those cells. Now down in Texas, I normally tell them this is a rod and this is a cone right over here. But being up here, we don't have to do that. and usually any other place, I don't have to either. So we would notice in that cell a membrane. And the membrane is folded up. Can everybody see that right here? It starts out and it's folded on top of itself. And the membrane is the photoreceptive part of the photoreceptor. And the membrane is what is going to capture the light. And you want to be able to capture a lot of light. And if you were to take the area of these membranes that are folded up in all of these little photoreceptors in the back of your eye, and you could take that membrane, unfold it, and spread it on the ground, for each of your eyes, it would cover a tennis court. Wow. Wow, that's a lot of light receptive material which is folded up neat and tiny in the back of your eyes so it's able to collect light. And other animals even have more. An eagle has even more in the back of its tiny eyes than we do. So you have a lot of light collecting capacity all folded up in these tiny little membranes in the back of your eye. And this is what it looks like magnified really, really big with an electron microscope, looking at that membrane which is folded up in one of those photoreceptors. Now what's cool about this photo is it shows that the membrane actually has two sides to it. Look here, really close. Can everybody see that this membrane actually has an inner and an outer layer to it? Where it's been pulled apart, there's an inner and outer layer of the membrane. So you're thinking, what goes on that membrane? Well, I'm glad you asked, because I'm going to tell you. It's really important what actually happens on the membrane. So if we were to take a tiny little portion of the membrane and blow it up, there is a process which is taking place on the back of it. The process takes nutrients that you eat, particularly one called vitamin A, also called retinal, and it brings it in and it undergoes a chemical reaction which will take that molecule and it will bend it. Bend it. Now that's important. As it bends it, it's putting energy into it. It's like you take a bow, which you're going to shoot a bow and arrow with, and you do what with the string? You pull it back and it bends the bow. You've put energy into the bow. You've loaded the bow. And once that molecule is bent, another enzyme is going to attach it to another molecule called opsin, which is what it happens right here. Here is the opsin with the bent retinal attached to it. The bent retinal attached to it. And it does all of this process in the dark. And what it's doing is it is loading up billions and billions of these molecules in this loaded state, ready to be fired when a photon of light hits it. So it's loaded, ready to be fired when a photon of light hits it. Now, how do you get from light to data in your brain? That's kind of cool. Well, this molecule is key to that, because here it is in this drawing. Here's the round opsin molecule. Here's the bent retinal molecule. And lo and behold, in comes a photon of light in the two layers of that membrane, which I showed you. Here are the two layers. One single photon of light hits that molecule. It makes the bent molecule go straight. Twing. You've just let go of the bow. The bow goes to its straight shape. Once it goes to its straight shape, it detaches from the opsin molecule. Once it detaches from the opsin molecule, it activates an opsin molecule. That activated opsin molecule will then activate dozens of another molecule called transducin. So it's amplifying the effect. Once transducin is activated, it will attach to another molecule called phosphodiesterase. Phosphodiesterase becomes activated, moves over, attaches another molecule which is holding a pore open. The pore is allowing sodium molecules to flow through that membrane in a chemical equilibrium. But once phosphodiesterase transfers over to this pore, it attaches to another molecule called cyclic GMP. It pulls cyclic GMP off of that molecule. The pore, which was being held open by cyclic GMP, slams shut. Sodium can no longer flow in at the same rate it is flowing out, creating a chemical imbalance, which creates an electrical charge across the membrane. And once it slams shut, your brain will send 70 millivolts of electricity as one data point back to your brain. Praise the Lord. One data point. Now, you can send signals with the data point. When I was a little kid, I had an older brother, and he was always getting into trouble, and I could send him signals. I could flash the light to the bedroom one time, Mom's coming. Flash the light twice, Dad's coming. 3 times, run for your life. You can send signals if you know how to interpret the data. Well, your brain is sending all kinds of data points with these hyperpolarizations to the back of your brain. How many are being sent? Here's the only real long quote in this whole talk, taken from a computer magazine called Byte. To simulate 10 milliseconds, 10 one-thousandth of a second, of the complete processing of even a single nerve from the retina would require the solution of about 500 simultaneous nonlinear differential equations 100 times, and would take at least several minutes processing time on a Cray supercomputer. Keeping in mind that there are 10 million or more such cells interacting with each other in complex ways, it would take a minimum of 100 years of Cray time to simulate what takes place in your eye several times a second. Wow. That is a lot of data being sent to the back of your eyes, and it is all being carried by the optic nerve. Now, the optic nerve is going to carry the data to the back of your eye, and here's a picture of it. Here are those eyeballs again. Here is the optic nerve coming from the back of the eye, and it's going to go off to the brain. Now, those are huge nerves. Huge nerves. If I were to poke you with a pin, the nerve that picks that up is microscopic. You'd have to have a microscope to see it. These nerves are massive. That's because there is essentially a one-to-one nerve association from these photoreceptors to the brain. So every photoreceptor has its own dedicated nerve sending a part to your brain. That's because you're taking in over 70% of all of your information visually. And that's why right now, in just one of your optic nerves, you are carrying more data than all of the telephone lines of New York City. That's how much data is flowing back to the back of your brain. Now, before I go off the slide, one last question. Is this brain from a man or from a woman? No, never mind. We'll skip that right there. All right. That was just a joke. Okay, so all of that data is now flowing from your eyeball to the back of your head. And it doesn't just get sent in a jumbled pattern. It is kept organized. So data coming from the right side of your body is kept organized with data coming from the left. which I didn't say the right eye and the left eye. Both eyes see the right side and both eyes see the left side. Both eyes see the top and both eyes see the bottom. Information from the right side is taken from both eyes, segregated together. Information from the left is taken from both eyes, segregated together. As it shows right here in this diagram, it is brought together. So information from the right side is brought over here to the left side of the brain, left side right over to the right side of the brain. It is carried through these optic tracts right here. This is looking at the bottom of a brain, back here to the back part of your brain, which is where vision is taking place. So if you take your hand, you put it right where your neck kind of touches the back of your head, that's where the vision is actually taking place in the brain. So now you know how light comes in, how it's turned into an electrical signal, which is data, which is carried by the optic nerve to the back of your brain. How do you actually see something? How do you see something? Well, seeing is a massive process of matching data with memories. Data is coming in and it is making a pattern. Your brain picks that up in the visual area and it sends signals off to other parts of your brain called association areas. Association areas where you have memories with stored data patterns. Stored data patterns. And when the data comes in, matches a memory that you have, you see something. And you get the perception of seeing things. So, in your brain, you have a memory of a man. You have a memory of a man who is moving his arms. You have a memory of arm moving. In your brain, you have a memory of a young, virile man moving his arms and that matches the pattern in your brain. And that wasn't a joke. I mean, that was one where you're supposed to say amen to that. All right. So, you're matching the patterns right now. And you don't even have to see things to match the patterns. For instance, you have this picture up here on the screen. It's not anything in your mind. But if I were to say to you, hey, what's up, Doc? Who do you see? You see Bugs Bunny. How many of you can see Bugs Bunny by just saying, hey, what's up, Doc? What color is Bugs Bunny? What's he got in his hand? Carrot, you see? It never fails. You all see it. That's because just hearing the words can match it to a memory, which then signals back to your visual area, which enables you to see it. Now, this is incredible because we can match patterns really fast. Like, what's that? Mountains, snow, green trees, blue skies, beautiful scenery. This is from my home state of Wyoming, where I grew up in. So we have all of these kind of pictures here. And look how fast your memory matched all of those patterns. Just try to get a computer to do something like that. What's this a picture of? Well, this is Texas. You know, this is these are mountains, and these are like big green trees that they would have down there in Texas. But immediately your memory matches a barren wasteland. And you know, as soon as you can see those kinds of things, it's very, very quick. Okay, so what is this a picture of? That's right. It's a turtle. Now this turtle kind of confuses you with these two little fake eyes on the back of his head. So what was cool about that is you looked, you didn't match a pattern, you looked again, got more data, you look again, and your brain went through this iterative process of trying to match the data until you saw, oh, that's a turtle. Now can everybody see the turtle's real eyes up there? Mm-hmm. Now you see those things. So what's this? That's a handful of algae. Algae, that's what it is. Man alive. That's a handful of algae right there on that. So, in your brain, you match sights with memories of things. And what's interesting is, from my perspective, as soon as the picture went up, the faces kind of went, ew, gross, and something like that. That's because memories that are tied to emotional experiences get laid down better. And so that's a handful of algae right there on the screen, which is invading some lakes and causing some problems there. So, what's that? A little stickworm right there. And he has his tiny little legs, and he's camouflaged, and he's hiding it. All right, let's wrap it all up in the next 2 minutes, and let's see how we integrate all of these things together. All of these things together right here. So, it's a picture of a baseball player. Are there any baseball teams in New York? All right, okay. There's baseball teams in New York. Let's say this guy is a New York Yankee, okay? And the pitcher is from the Boston Red Sox. All right, and the Boston Red Sox, New York Yankee. Even a Boston Red Sox pitcher can throw a fastball of what? 90 plus miles an hour. So he's going to throw that fastball. The pitcher is going to hit a home run because by definition he's a New York Yankee. So he's going to swat it out of the park right there. The batter on that. So what has to happen? What actually has to happen here in order to do that? First of all, light has to come from the sun or stadium lights. It has to hit the baseball, be reflected off the baseball, be brought into the back of the eyes. It's sent all the way back to the visual center. It's sent out to association areas where the batter says, this is a baseball. And maybe since he's a real good batter, he already has a memory of what type of pitch it is, a curveball, a slider, or something like that. All of that information is then going to be sent up to the frontal association area where not only does he decide what type of pitch it is, he's actually going to make a decision, swing or not to swing. And let's say he's going to swing at that ball. So he makes the decision to swing at the ball. What does he do again? He regathers additional data from that. That data is actually sent to an area of the brain, which isn't even marked over here, called the premotor area. In the premotor area of the brain, this batter has stored memories of all of the gross movements, how he's going to turn his body, how he's going to move his arms, and all of those kinds of things. That data is matched to the incoming visual data. It is sent back down to another area of the brain called the cerebellum. Now that's the part of your brain which enables you to balance and do fine motor skills so that you can actually swing the bat and not fall flat on your face. So that information is going to be combined with the premotor information, and that package of information is going to be sent up to the motor part of the brain. Now the motor part of the brain is where the batter is going to make fine movements, such as how he's going to shift his weight from one knee to the other, where he's actually going to turn his knee or not, how much he's going to grip that bat, whether he's going to flex his wrists in some other way or another way. That information is packaged down, updated with additional information coming in from the ball. Because he's a good batter, he keeps his eye on the ball constantly, and he's updating it, and the brain is constantly going through this loop on that. Finally, it is all packaged together, sent down the motor area, sent down the spinal cord. The signals are sent out to his neck, to his trunk, to his arms, to his legs, all the way down to his toes. He swings the bat, it connects with the ball, and he pops it out, and he makes a home run. And he does all of that in much less than a second. And he pops it all out. Now that is pretty amazing. And he does it all through this visual system. And every time we see a batter actually pop one of those balls out, we should say, I will praise you because I am fearfully and wonderfully made. Oh man, anatomy, which fits like a hand in a glove. I knew someone would get a chuckle out of that. Sorry. It does. It fits like a hand in a glove. But anyway, it all fits together. And when people start seeing how things fit, they know they only fit by a design source. So you use the word fit. Birds' wings fit their environment. Fish gills fit their environment. These things fit. And the things we're going to talk about on the eyeball are fit. And when people start hearing how things fit, they will start praising the Lord because they'll know that they are fearfully and wonderfully made. Well, this is what we're going to talk about. This is a painting of a cross-section through an eyeball by this man named Frank Netter. He was also a medical doctor, but he was a great artist. And everybody who goes through medical school studies Frank Netter's pictures. And this is a cross-section through an eye. You can see your cornea right up there. You can see your lens here. You see the iris with the pupil there, the retina in the back. We're going to talk about all of those things. This man is now dead. I heard him interviewed on ABC News by Peter Jennings, who's also dead, before he died, and I don't know whether he was a believer or not, but he said in his interview, I find it hard to believe that all the things I've been painting for all these years could have come about by chance. And that just popped out. I'm surprised ABC didn't edit it out of the interview, but it was in there just to say. I want to give him credit because I'm going to use some of his pictures. Now, visual system, light. Dr. Mason already mentioned this word, photon. A photon is the smallest unit of measurable light. It may be a particle, it may be a wave, it may be a wave-like particle. I don't know, but they travel at obviously high speeds, which is the speed of light. There we go, traveling at the speed of light. You see various colors reflecting off the screen, and those are variations of absorption. And light is just pure energy, and you're able to detect the smallest unit, a single photon of light. And what we're really after here, since it's a designed mechanism, is we need something that actually has an ultimate purpose. And the purpose is to get information. That's what it really means to see, is to get information. And so I'm going to mention that over and over again. The purpose is to eventually store information. So you need a mechanism which is able to sense, detect something. Light is reflecting off this screen. It's coming into your eyes. You have a detector mechanism that is called your eyeball that detects the light. And then you need another system to make sense of the data and convert that into information. That is your brain. You have a visual system which consists of your eyeball and your brain and all the muscles and everything which make those things work. This is not a talk on the eyeball, which everybody believes that it is. It's on the entire visual system and how these things must work and function and operate together. And I use the word miracle because it is a miracle, because it's going to show that all or nothing unity in which all the parts must be there as nothing's going to function. Well, the development of the visual system, there are two explanations. Supernatural creation, all of these parts fit together for a purpose, i.e. all the features of design because it had a real designer. Or the naturalistic evolution explanation, in which a series of random genetic mistakes were guided by natural selection, whatever that is, culminating in a highly, today's topic is, behold his beauty, design of the human visual system. So, if you're coming to this talk, we have to use some words in this talk. So is everybody comfortable with the word eyeball? Okay, eyeball. All right. And then you can, and we're going to look at some pictures in this, and we're going to look at some pictures and features of the eyeball. And for some reason, some people get freaked out when they look at an eyeball up close and stuff like that. It's like, oh, I can't stand it. So if you're looking at a picture of an eyeball or a little bit of a segment of an eyeball, and you get that queasy, woozy, quoozy feeling, please vomit sideways, not forward, on me, and share the love with the person next to you on that. After Dr. Perdam's lecture, you'll just be sharing some symbiotic bacteria with them on that. Designing the human visual system. Well, remember this hypothesis. Worship should be the normal response to science. Hasn't changed. We're fearfully and wonderfully made, and now we all know to say the whole verse, I will praise you because I am fearfully and wonderfully made. Everything is there is made. Made. Boy, pound this word into your mind. Made. The Lord has has disclosed himself in Romans number 1 by the things that he has what? Made, even his eternal power and Godhead, so that they are without excuse. I wish we had never lost the words intelligent design, because everything is a design argument. And please, please, let me encourage you, start looking at your human body like you would look at any other sophisticated designed entity. You can only read a complex system. Those are going to be the two explanations. You decide at the end of this talk which one of these best fits the data. Well, one person, his name is Richard Dawkins. Man, what a treasure trove of quotes for us, because he states what's on his mind very candidly. In a book called The Blind Watchmaker, he was lamenting that people have a hard time understanding evolution. He says, it's almost as if the human brain were specifically developed to misunderstand Darwinism and find it hard to believe. Wow. You know, it's like that, because the Bible says these things are clearly seen right there. The conclusion that our brains seem predisposed to resist Darwinism stems from our great success as creative designers. Our world is dominated by feats of engineering and works of art. We are entirely accustomed to the idea that complex elegance is an indicator of premeditated design. And to that I would say, no duh. You know, when we see complex design and we conclude there was a designer, that is a totally reasonable, totally rational conclusion. It's the irrational conclusion which sees complex design and believes that's all an illusion of a designer. He goes on to say, this is probably the most powerful reason for the belief held by the vast majority of people that have ever lived in some kind of supernatural deity. The Bible is true. It is clearly seen, and people in any age and any generation see it. Of course, Dawkins thinks that the vast majority of people that have ever lived are all wrong, and he is right. But we're going to see that the vast majority of people are probably right. Well, your eye is going to begin development about the 4th week of pregnancy. Really understand it as you understand the principles of design. Oh, we need to get the rest of our creationist biologists on board with this. You can only understand it properly as you understand how things are designed. So we're going to look at some of the functions of design, and if you want to write it down, it's nowhere on the slides, but designed complexity is this. Hold up your hand. It is multiple parts working together for a purpose. That's what complexity is. Multiple parts working together for a purpose. We could put a mathematical probability on this. We could do that. But multiple parts working together for a purpose is biological complexity, and that has only been observed in any other area from an intelligent source. Natural forces alone, gravity, wind, rain, all those things can't even produce 2 parts working together. Not to mention all of the parts we have working together. So biological complexity is what? Multiple parts working together for a purpose. And you know what? When people see multiple parts working together for a purpose, inside their mind is saying designed, designed, designed. And the next word that you want to use when you're talking with them, this is all extra, it's not even a talk, is the word fit. Fit. Things only fit together when they are designed. It's the perfect fit of things which shows the evidence of design. Now, I talked about human reproduction today. Did everything, even at the cellular level, just fit perfectly? It fits like a hand in a glove. And we didn't even talk about the external